Today's speaker is Umnia Doha. She's been working with us for not quite a year, and she graduated with a PhD from Saif's lab over in engineering. Next week, Kingsley's going to be, no, not Kingsley. Next week, Austin's going to be speaking to us about the X5000. And so that's an X-ray CT system, and if you know anyone in engineering or biology that wants to do X-ray of their samples, that would be great. Today, Umia is going to tell us about our Steadicon system. It was installed a couple months ago, and based on the title, you can see that it works both as a confocal and also as a 40 nanometer STED system. So welcome.
Okay, thank you, Glenn. Welcome everyone to my presentation. Today I'm going to introduce the Steadicon, which is the latest addition to our list of confocal systems. It also has the additional super resolution capability. Okay, so okay, now it's working. Okay. So in terms of super resolution capabilities at the core, we have the SRSIM and the LSM880 with the air scan detector, which gives you a resolution of 140 nanometer. So when we are talking about super resolution, we are talking about a resolution limit that's below the diffraction limit of light, which is 200 nanometer. Then we have the LSM900 with the deconvolution software, which allows you to go down to a resolution of 90 nanometer. And then we have MinFlux, which gives you a resolution of 2 nanometer right here. So Steadicon falls somewhere between these two, which has a resolution of 30 nanometer. So if you have a sample that you are interested in a structure that has a size limit around this range, so this could be an ideal microscope for you. This is what the microscope looks like. So this is the Steadicon unit right here. This is from Aberrior. It has 4 excitation lasers, just like any other confocal microscope we have. In addition to that, it has a 775 nanometer STED laser, which gives you that extra resolution. It is attached to this inverted Nikon microscope base. It has all the magnifications just like any other confocal has. And so this is capable of doing 3-color 2D STED, meaning that you would get that super resolution on your XY plane, but your Z resolution would be still as same as the confocal resolution. So yes, it's a 3-color 2D STED. As I mentioned, any STED microscope is also a confocal microscope. So even if you don't need that extra resolution, you can still use it as just a regular confocal.
Umnia, you're on mute.
Oops. Okay. 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 Yeah, I think it is. Yeah, I think. Okay. Okay, sorry about the Zoom people out there. So, okay, I just got started. So we are talking about the STED system we have here in the course. So it is a 3-color 2D STED system. You can do a regular 4-color confocal microscopy with this one as well. This has 4 APDs, single photon counting detectors. So these are much more sensitive than the photomultiplying tubes we have on the other confocals. It has the autofocus capability, which allows you to do long time lapse and has that drift correction system. You can also do large area scan and multi-positioning different region of interest with this microscope. Okay, so let's go to some of the examples. So this is an image of synapses of the neuromuscular junction of a Drosophila larva. So this is a confocal image, so you can make out the structures, but it doesn't give you any information about the synapses. After you do STED, so you can kind of see the puncta-like structures, so you get that extra information. Here is another example of the two neuronal proteins, pectrin and adenosine, in rat hippocampal neurons. So from this confocal image, you can kind of guess there are two proteins, but you don't have any information about their organization. After doing STED, you can see the periodic structure of these two proteins. So this is what STED enables you to do to get that super resolution allows you to get those information which confocal doesn't allow you. Okay, so for this presentation, I'm gonna cover the basic principles of how STED work, some tips and tricks for sample prep, and finally some user applications that we have got so far. Okay, so before I start talking about how STED works, I just want to briefly go through the diffraction limit of light. So as I mentioned before, super resolution, when we say super resolution, it's the resolution that's below the diffraction limit of light, which is 250 to 200 nanometer. So in a microscope, in an ideal case, if you have an object, you would expect an image which would have the similar size as your actual object. But in reality, because of the diffraction limit of light, you see this giant blob instead of, if your object is very tiny, you wouldn't be able to resolve that because of the diffraction limit of light. So if you have two objects, you would still see this. Now, this is fine if they're more than 200 nanometer apart, but as they get closer and closer, it becomes harder and harder to resolve them. So that's when STED comes into play. So in a regular microscope, you'd have your object, you'd excite it, and then the fluorescence emitted by the object is then detected here, which is, again, diffraction limited. So when we are trying to do STED, we introduce another laser here, which is the depletion laser. So in addition to the excitation, you have this depletion laser, and with some optical engineering, you have this donut-shaped laser, which is aligned with the excitation laser right here. So what this donut-shaped laser does is it would overlap with the excitation, and it would, any fluorophores that from the emission of the sample, which are overlapping with this annular region of the donut, they would be depleted with this depletion laser, and you would only have the signal that is in the center. So that's how you're getting rid of all those fluorophores around here, and you are getting a tighter spot at the center of your sample. As you increase the depletion laser power, you make this spot smaller and smaller, and that's how you get you detect a smaller and smaller spot, which allows you to get that extra resolution that you get from STED. Okay, so let's talk a little bit about the physics behind it. So we use a Jablonski diagram to explain this. So what happens here is when you have a fluorophore, In fluorescence, you would excite it with a laser. It would go to a higher energy state. Then it stays there for maybe a few nanoseconds, and then it would relax, go down to the ground state, while it will emit a photon, which we see as fluorescence. So this is a spontaneous emission, which we can term as fluorescence on. Now, when we are doing STED, we, in addition to the excitation laser, after exciting it, while the fluorophore is still on the higher energy state, we hit it with the STED laser, this donut-shaped laser right here, and then force this fluorophore to go down to the ground state. So instead of the spontaneous emission here, we are forcing it to go down to the ground state. And as we know the exact wavelength where it's going to emit, because we are stimulating it, we can turn the fluorescence off and filter it out. So that's how we get a fluorescence on-off switch, which we can then use with our system to get the STED system to work. So this is just a brief description of the physics behind how we are getting that super resolution capability instead. This is just a regular schematic of the light path. So you have the objectives, you have your excitation laser, STED laser passes through the objective, aligned together. So that's how you detect your fluorescence. Now, the STED laser is also tunable, meaning that you can increase or decrease the STED laser power, like I said before, and basically control the diameter of the center and get higher and higher resolution. So here I have some 40 nanometer red particles, and you can see that we get the point spread function right here. So as I increase the STED laser power, you can get a higher and higher resolution until you reach the nominal diameter of 40 nanometer. Okay, so from that, let's move on to some of the sample prep techniques. So you can apply for your samples. So I want to start with saying that any existing confocal protocol that you are currently using, it can be modified into a STED protocol. So it's not some very widely different techniques that you have to apply. It's slight modifications would be just fine. One of the first thing you can do is we always, if you have the option, choose an organic red dye if it's just a single color STED. If you're planning to do a two-color STED, choose a red plus orange dye. So the reason behind this is, if we look at the emission spectrum, so this is your 770 nanometer STED laser. So it has to overlap with the emission spectrum of your dye. So that is the reason. So since it's redshifted, so that's why you want a redshifted dye that would overlap here, and you can see the reason why we prefer a red dye over an orange dye, because the overlap is higher here in this region. So if you can, please go ahead with a red dye instead of an orange dye. The second, if you can avoid DAPI, please do so, because DAPI is very bright, and even if on the other channels, sometimes it has it just induces a background, and then it becomes hard to deplete those with the STED laser. So if you can avoid DAPI, go ahead. If you have to use it, please use as low concentration as possible. Also, DAPI can also get 2 photon excited by the STED laser, so that has another effect as well. So yeah, if you have to use DAPI, go as low as possible, and please keep an eye on your mounting media. Some mounting medias have DAPI in it. Avoid those for your STED. For glass cover slips, it's recommended to use number 1.5 because the objectives are configured for that cover slip. So you might think number 1 is is thinner, so just use that, but no, go ahead with 1.5. And for three color sted, if you have to use a 3-color sted or a green dye, you can't do a sted because of this reason. So your regular blue or green dye, the emission spectrum doesn't have an overlap with our sted laser. So you can use a long stock shift dye. In this case, like Aberrier has this dye, which has, I think, about a 90 nanometer stock shift. So even if the excitation laser is green, your emission spectrum overlaps with the sted laser, so you would be able to do green channel sted. Okay, so next, let's go to live cell imaging. As I mentioned earlier, it has an autofocus, so it allows you to do long time lapses, and you can also do it with live cells. So here is a video of live cell. I think this video was taken for 8 hours. The green is labeling the cristae of mitochondria, and I think the magenta is endoplasmic reticulum. Here they used a dye called PK Mitored. So this is, it directly tags the mitochondria on these cells. There are some dyes available commercially which can directly label your protein of interest. For example, actin and microtubules have dyes that you can use for doing live sted. Now, if you don't have a dye for your structure of interest, you might be tempted to use fluorescent proteins. So that's what we mostly use when we are doing live cell imaging. But it's highly recommended not to do so, because for doing sted, since we are depleting many of the fluorophores, we want a bright and photostable dye. And fluorescent proteins are not that stable. So that's the reason it's recommended to use organic dyes over fluorescent proteins. And so if you don't have the dye for your structure, you can express SNAP or halo tags in your sample and then use silicon rhodamine dye, for example, to label your sample. So here is a list of dye recommended by Aberrier for doing sted. On the left, I have the dyes for fixed sample. On the right, these are the dyes recommended for live cell imaging. So one thing I do want to mention here is the green dye that's recommended here are for a depletion laser of 595 nanometer, which we don't have in our system. So if you're planning to do a 3-color sted or a green sted, use the long stock shift dye that I just showed before. Okay, so yes, I guess that was all. There are, however, more information on sample prep. If you go to Core Facilities website, look up on the MinFlux workshop videos from last year, you can find a lot more information over there. Okay, so let's look into some of the applications we have done so far with our users. So this is an image of speckled proteins in fibroblast cells. Thanks to Minju from Prasant Lab, she made the samples and imaged those. So the green is a protein SON, and the purple, the magenta is the SPRM2. So these are two nuclear speckled proteins, and this is a confocal image, so you can kind of make out their structure, but again, it's not giving you precise information about their size or shape. So after we do sted, this is how it looks like. So here you can see, let me just do this. So you can, it's hard to see the magenta one, but you can still see their position relative to each other, the green and the magenta one. So so it gives you information about their relative position, which you don't get when you are doing just confocal. Here is another example from Van der Donck's lab. They are looking into a small peptide protein, so which is tagged by the Aberrier star red here. On the confocal, you just see a blob like others. If you do a STED, you can see a finer structure. So this project is still ongoing. I think the goal is they're going to use another small peptide protein, label it with a steadable dye, and look at the localization between these two peptides. And here again, there is DAPI and green is actin, so you can do multicolor confocal. It's just that you won't be able to do STED. But yes, you can use it alongside the STED just for confocal imaging. Yeah. Okay, so like I said, you can do just regular confocal. This is a 4-channel confocal image. You can scan large areas with this system. Then you can also select region of interest and scan them. Because of the autofocus, you can select these areas, do an overnight scanning, and then get long time lapses as well. Okay, so next, let's compare some of the data from Steadicon with our LSM 900, just to get an idea of the resolution we are gaining. So here is an image of nuclear pore complexes labeled with a barrier star red. On the left, I have the confocal image taken with an 100x oil immersion objective. On the right, I have the STED image. You can clearly see it's resolving the much finer structure with the Steadicon. And then we use the same sample to image in our LSM 900 system. So here is the raw data of the same nucleus. This was taken with a 63x oil immersion objective, I believe. And then after you do the ARIScan processing, this is how the sample looks like. And this is finally what it looks like after you do the deconvolution in LSM 900. So now let's just select a small region here and then compare it with our STED. So this is how it looks like. This is your image in the 900 after deconvolution. This is your STED. And this is the data of the point spread function of this specs right here. It's the full width half max. Here we measured those for multiple of these specs here and then got the data. So you can clearly see how STED is giving a much higher resolution compared to the LSM 900. So this is kind of visual comparison between the resolution of the two. While we are at it, you can also compare the calendar between the two. So this is in February, so you can see how the LSM 900 looks like, and you can see this is particularly empty. So yes, again, if you are doing confocal, if you're having a hard time using 900 or any of the other scopes, this is free, you can just always use it. So this can be another incentive for you to use the microscope if you want to. Okay, so, and then I think, what do I have else? Finally, let's compare some data with MinFlux since we are doing the comparison. So as I showed before, MinFlux has a resolution of 2 nanometers. So this is the same image of the nuclear speckle protein from Pasant lab. This is the nuclei, and if you can focus in one of the speckles right here, and after you do the MinFlux here and do the reconstruction, this is the image that you get, and the corresponding STED image taken with Steadicon is right here. This is the same protein targeted here, and then if you zoom into one of these speckles, this is just a zoomed in view, this is how it looks like. You can still resolve individual specks, and we also did a measurement of the full width half max point spread function of each of these, and it's approximately 40 to 45 nanometer. So yes, if you have a sample, a structure of interest that falls in this resolution limit, Steadicon might be a very good system for you to use. Okay. Here is again a side-by-side comparison between the two. Here is your MinFlux image. Here is the Steadicon image of the same sample. Okay, so finally, as a summary, this system, the Steadicon we have, it is a confocal system in addition to the STED system. It can do all the 4-color confocals that one would commonly use, so you can use it for that purpose. If you are doing STED, just remember that you would only get the super resolution in the XY plane. Your Z plane is still limited by the diffraction of light. The resolution can go down to 30 nanometers. So just as I mentioned before, it falls somewhere between LSM 900 and MinFlux. So even before you are getting started with MinFlux, you can use this system as a quick check if how good your what resolution you are getting, and then you can move for a higher one. And finally, with some tips and tricks, you can prepare your sample for live cell imaging. You can do some large area scan, and with the autofocusing capability, it allows you to do long time lapses for live cell STED. We have these videos, like I just mentioned, in the core website from last year MinFlux workshop, where you have more information, especially on the sample prep part. That might be very helpful if you're just getting started with making samples for prep for STED. Okay, so yeah, I think with that we can end and open for any questions.
For Umia?
Yeah.
How much does it cost to use this compared to others?
Find that information on the website. I don't have it.
Not on there.
It's not on there.
That's the one that's not listed.
Okay.
$10 more.
I don't have it on top of my
Yeah,
mind.
it's close to the $10 more. I'm not sure. I should know that, I guess. I was just looking at it this morning.
Can I ask what the time difference is between taking like the MinFlux image and the STED image?
So the STED image, the sample I just showed, the speckled proteins, I think it took few minutes, 10 to 15 minutes for that one sample. For MinFlux, I think it's much longer. I don't know the exact time it took.
An hour, 2 hours for that
Yeah.
answer? Yeah, so it's a few minutes compared to a couple hours.
Fair enough.
Typically 1 hour for a class.
No questions?
If there's no other questions, let's thank Umia again. As you're thinking about what you're going to use, remember the calendars and make sure.
Yeah.
So if you don't want to work at night or whatever, I think the Steadicon will work great for you. So anyway, thanks for coming. Remember, next week we have an X-ray CT talk from Boston.
Okay.
There's a little bit more pizza left.